Hello, so today we are looking at cells involved in immune responses and antigen recognition. The immune system is developed to protect the host from pathogens and other foreign substances. Non-self discrimination is one of the hallmarks of the immune system. There are two main sites where pathogens may reside: extracellularly in tissue spaces or intracellularly within a host cell, and the immune system has different ways of dealing with pathogens at these sites. Although immune responses are tailored to the pathogen and to where the pathogen resides, most pathogens can elicit both an antibody and a cell-mediated response, both of which may contribute to getting rid of the host pathogen. However, for any particular pathogen, an antibody or cell-mediated response may be more important for defense against the pathogen. So looking at extracellular pathogens, antibodies are the primary defense against extracellular pathogens, and they function in three major ways: neutralization, opsonization, and complement activation. During neutralization, by binding to the pathogen or foreign substance, antibodies this can block association of the pathogen with their targets. For example, antibodies to bacterial toxins can prevent the binding of the toxin to host cells, thereby rendering the toxin ineffective. Similarly, antibody binding to a virus or bacterial pathogen can block the attachment of the pathogen to its target cell, thereby preventing infection or colonization. With regards to opsonization, antibody binding to a pathogen or foreign substance can opsonize the material and facilitate its uptake and destruction by phagocytic cells. The Fc region of the antibody interacts with Fc receptors on the phagocytic cells, rendering the pathogen more readily phagocyzed. Complement activation. Activation of the complement cascade by antibodies can result in lysis of certain bacteria and viruses. In addition, some components of the complement cascade, for example C3b, opsonize pathogens and facilitate their uptake via complement receptors of phagocytic cells. Let's move on to intracellular pathogens. Because antibodies do not get into host cells, they are ineffective against intracellular pathogens. The immune system uses a different approach to deal with these kind of pathogens. Cell-mediated responses are the primary defense against intracellular pathogens, and the approach is different depending upon where the pathogen resides in the host cell, for example in the cytosol within vesicles. For example, most viruses and some bacteria reside in the cytoplasm of the host cell. However, some bacteria and parasites actually live within endosomes of the infected host cell. The primary defense against pathogens in the cytosol is the cytotoxic T lymphocyte. In contrast, the primary defense against a pathogen within vesicles is a subset of helper T lymphocytes called Th1 cells. Cytotoxic T lymphocytes are a subset of T lymphocytes that express a unique antigen on their surface called CD8. These cells recognize antigens from the pathogen that are displayed on the surface of the infected cell and kill the cell, thereby preventing the spread of the infection to neighboring cells. These kill by inducing apoptosis of the infected cell. T helper cells are a subset of T cells that express a unique antigen on their surface called CD4. A subpopulation of Th1 cells are the primary defense against intracellular pathogens that live within vesicles. Th1 cells recognize antigens from the pathogen that are expressed on the surface of infected cells and release cytokines that activate the infected cell. Once activated, the infected cell can then kill the pathogen. For example, Mycobacterium tuberculosis, the causative agent of tuberculosis, infects macrophages but is not killed because it blocks the fusion of lysosomes within the endosomes which resides. Th1 cells that recognize Mycobacterium tuberculosis antigens on the surface of an infected macrophage can secrete cytokines that activate macrophages. Once activated, the lysosomes fuse with endosomes and the Mycobacterial tuberculosis bacteria are killed. Although immune responses are tailored to the pathogen and to where the pathogen resides, most pathogens can elicit both an antibody and cell-mediated response, both of which may contribute to ridding the host of the pathogen. However, for any particular pathogen, an antibody or cell-mediated response may be more important for defense against the pathogen. Now let's move on to looking at cells of the immune system. All cells in the immune system originate from a hematopoietic stem cell in the bone marrow, which gives rise to two major lineages: a myeloid progenitor cell and a lymphoid progenitor cell. These two progenitors give rise to myeloid cells, monocytes, macrophage dendritic cells, megakaryocytes, and granulocytes, as well as lymphoid cells, T cells, B cells, and natural killer cells, respectively. These cells make up the cellular components of the innate, non-specific, and adaptive specific immune systems. Cells of the innate immune system. Cells in the innate immune system include phagocytic cells, monocyte macrophages, and PMNs. NK cells, basophils, mast cells, eosinophils, and platelets. The role of these cells have been discussed previously, and the receptors of these cells are pattern recognition receptors that recognize broad molecular patterns found on pathogens. Now looking at cells that link the adaptive and innate immune systems, a specialized subset of cells called antigen presenting cells are a heterogeneous population of leukocytes that play an important role in innate immunity and also act as a link to the adaptive immune system by participating in the activation of helper T cells. These cells include dendritic cells and macrophages. A characteristic feature of APCs is the expression of a cell surface molecule encoded by the genes in the major histocompatibility complex, referred to as class 1 MHC molecules. Class 2, B lymphocytes also express class 2 MHC molecules, and they also function as APCs, although these are not considered as part of the immune system. In addition, certain other cells, such as thymic epithelial cells, can express class 2 MHC molecules and can function as APCs. Cells that make up the adaptive immune system include the B and T lymphocytes. After exposure to antigen, B cells differentiate into plasma cells whose primary function is the production of antibodies. Similarly, T cells can differentiate into T cytotoxic or T helper cells, of which there are two types, Th1 and Th2. There are a number of cell surface markers that are used in clinical laboratories to distinguish between C cells and their subcells populations. Let's have a look at a bit more detail. So, main distinguishing markers of T and B cells. You can see here different types: B cells, T cytotoxic, T helper, and what is present and what is negative. CD3, CD4, negative, CD8, positive, and T cytotoxic. They've got the different antigen receptors, the BCR surface antigen, the TCR, and the TCR, respectively. So let's look at lymphocyte recirculation. So since there are relatively fewer T or B lymphocytes of a receptor for any particular antigen, the chances for successful encounter between an antigen and an appropriate lymphocyte are slim. However, the chances for a successful encounter are greatly enhanced by the recirculation of lymphocytes to the secondary lymphoid organs. Lymphocytes in the blood enter the lymph nodes and move through the lymph nodes. If they do not encounter an antigen in the lymph node, they leave via the lymphatic and return to the blood via the thoracic duct. It is estimated that 1 to 2% of lymphocytes recirculate every hour. If the lymphocytes in the lymph nodes encounter an antigen which has been transported to the lymph node via the lymphatics, the cells become activated, divide, and differentiate to become transported to the lymph node via the lymphatics and become a plasma or Th or Tc cell. After several days, the effector cells can leave the lymph nodes via the lymphatics and return to the blood via the thoracic duct and then make their way to the infected tissue site. Non-specific and specific. So in non-specific, the system in place prior to exposure to antigen, it lacks discrimination amongst the antigens and can be enhanced after exposure to antigen for effector cytokines. If specific, it is induced by antigen, enhanced by antigens, and shows fine discrimination. The hallmarks of specific immune system are memory and specificity. The specific immune system remembers each encounter of a microbe or foreign antigen, so that subsequent encounters stimulate increasingly effective defense mechanisms. Specific immune response amplifies protective mechanisms of non-specific immunity, directs or focuses these mechanisms to the site of antigen entry, and this makes them better able to eliminate foreign antigens. There are two main lineages that derive from the hematopoietic stem cell: the lymphoid lineage, T lymphocytes, B lymphocytes, and natural killer cells, and the myeloid lineage, the monocytes, macrophages, Langerhans cells, dendritic cells, megakaryocytes, granulocytes such as eosinophils, neutrophils, and basophils. Let's look at clonal selection. The four basic principles of the clonal selection hypothesis. Each lymphocyte bears a single type of receptor with unique specificity. Interaction between a fallen molecule and lymphocyte receptor capable of binding that molecule of high affinity leads to lymphocyte activation. Differentiated effector cells derived from an activated lymphocyte will bear receptors of identical specificity to those of the parental cell from which that lymphocyte was derived. Lymphocytes bearing receptors specific for self molecules are deleted at an early stage in lymphoid cell development and are therefore absent from the repertoire of mature lymphocytes.